Okay, so now I'm going to unfortunately take the chat off because I've got to move the screen, and I've got to move this to a full screen now. And we're going to go into the concept map itself. So this is a suggestion to you on how you can go about constructing your concept map. I'm not sure why I've got two stray arrows there, but in any way, let's get rolling. So the first thing you would want to have on your concept map is the boxes that will relate to the things that are actually on your slide. So make sure they're color coded, because this is essentially how the marker will know what's what. So you can choose any color you wish, but just try to ensure that the color you do use can be seen. So yellow with a white text is very difficult. That's one color we've found very hard to read the text behind it. So try to use colors that are fairly obvious and stand out from each other. Okay, so the first thing that we want to probably put on the map is what, guys? What would you think would be one of the first things that would populate your concept map? Yep, etiology. So the etiology for type 1 diabetes, we've already know, so it's an autoimmune dysfunction. So this is essentially the immune system not functioning correct and it attacking your own body. Now, you will have to have a bit of an understanding with your disease what that actually means and how you could express this in your own words. Okay, so the autoimmune dysfunction is essentially going to cause an autoreactive T cell. So your T cells is part of your adaptive immune system. You learned that in PMP1. So the T cells coordinate, particularly the T helper cells, coordinate the immune response, specifically the adaptive immune response. And what happens in these individuals is that self-tolerance. So they lose the ability to be able to tolerate their own cells, which are problematic, because if you do that, you're going to start attacking your own body as you would do if it was a bacteria or a virus. So what that would then do is produce antibodies. We know what antibodies are. These are things that are produced against an antigen. But unfortunately, this is produced against your pancreatic B cells. So remind me, students, what do the pancreatic beta cells do? Now, you should have covered this in A&P. So the pancreatic beta cells, what do you know they do for you in your body? What's their function? Yeah, they release insulin, right? So if you're producing antibodies against your beta cells, that's going to be somewhat problematic. So that's going to start to destruct, start to break down, start to kill off your pancreatic islet cells, which is essentially those cells, and that's the beta cells. So you're going to start to kill off those. Now, as you kill those off, you're going to start to cause inflammation, which is the itis, so insulitis, which is just another name for basically killing off the beta cells and having significant amounts of inflammation in that area. Now, as you start to lose your beta cells, you're going to have a drop in insulin production until there is a deficiency. And this is the kind of, what I've done here is the second criterion. This is set up your pathophysiology. This is a sequential process that would happen in time. So for instance, you wouldn't have insulin deficiency at the top and then have the antibodies following it, and then you have the self-tolerant loss there, because that wouldn't make sense. You have to have it in a sequence that actually would happen in time in respect to this disease. So we can almost finish this point at insulin deficiency, because that's going to be allowing you now to set up your clinical manifestations pretty well. Okay, so in terms of that process, does that all make sense to you? How we've kind of gone from the etiology, the cause, and we've gone through a stepwise process to a point that you think, well, this is going to be a point where a lot of my clinical manifestations will start to stem out of. You will find something similar with yours. So your etiology, you know, is your clots within your veins of your leg, and then you know that your diagnosis is a PE, so it's pulmonary embolism. So somehow you've got to demonstrate a process like I've just done, going from clot in the legs to clot in the lungs. So you need to demonstrate that process. Now I'm going to come back and address my risk factors now. So we know one of the risk factors was the type 1 diabetes of the father. And so now you have to come up with, well, why is this actually a risk? So you need to come up with a mechanism, a process. Okay, it doesn't have to be too much, but just so we can see that you've understood why this indeed is a process to increase the risk of it happening. So a type 1, having a positive family history, it puts a person into a genetic predisposition. So they're more likely to get this autoimmune dysfunction from a genetic basis. What's the genetics? Well, it's this gene here, the human leukocyte antigen gene. Okay, so I showed you that on the best practice. So that's just you going to the literature to try to figure out, well, what is that genetic predisposition? And having this predisposition is going to essentially lead to the autoimmune dysfunction. Okay. Michael, you need to use the literature to demonstrate where the risk factors fit in. Okay, so there could be sometimes risk factors will fit nicely into the cause, but sometimes, like we saw in PNP1, some of the risk factors were more associated with the pathophysiology itself. So for instance, with the breast cancer case that we did in PNP1, the younger age is not a risk for breast cancer, but it is a risk for metastasis once you get breast cancer. So the risk factor for breast cancer is actually an older age. A 50-year-old female is actually at a higher risk than a 30-year-old of getting breast cancer. So the 30 years age was not a risk to the breast cancer, but it was a risk for it metastasizing to the brain. So that's an example of where sometimes risk factors might not fit neatly to the cause. Okay, moving on so we don't run out of time. The next is, in her case, was celiac disease. So celiac disease is a disease that we know affects the gastrointestinal tract. So this actually becomes an environmental trigger. So it triggers the body environmentally to lead to then an autoimmune dysfunction. So it's not completely known how this triggers it, but it is known as an environmental trigger. Another one in her risk is her background, her Finnish background. Now this could be, so I've actually demonstrated here, there's 2 places that you could fit this in. So you can have more than one location if you think it can fit. So having a Finnish background from Finland, there is the genetic predisposition, which we're not exactly sure what the genes are, but there is a predisposition there, which is similar to what we saw with the father, but also the location. So persons that are located towards the ends, so this would be latitude based at the poles of the planet. So Canada, Alaska, Finland, Norway, are risks, and it's thought that there's something to do with sunlight. Maybe not as strong now with vitamin D. So vitamin D is maybe starting to get taken out of this, but there could be something to do still with the excess of sunlight, particularly in the winter months, that increase the likelihood of autoimmune dysfunction. Okay, so that's the risk factors done. Now what we can do is build the clinical manifestations out of it. So you won't be marked down on the direction of your arrows, but you will lose mark. Sorry, but you will. It's probably makes more sense to bring your risk factors in and your clinical manifestations out, because they come out of the process and the risk factors come in. But you won't be marked down if you have the arrows going in the other direction. Okay, so once we have insulin deficiency, what can't we do? Well, glucose can't enter your cells. So if you can't get glucose into your cells, you can't make ATP. So what would you expect to do? What would you expect to see in a patient if you're not making ATP? We know that ATP is all about energy. So what would you expect to see in a patient? Yes, that's great. Melissa, well done. So lethargy. So there's the first clinical manifestation. So just fairly simply, we've just demonstrated that just by not having insulin, you can't get glucose into your cells. Therefore, you can't make energy very efficiently. Therefore, you're lethargic. Okay, moving on. Now, once you start to not process this metabolism very well, your liver kicks in because its liver tries to look after your whole body by doing two main things to try to get your glucose levels back up, even though they're already high. But in this metabolic state, the liver thinks that you're having problems making energy, therefore your glucose levels are low. So it tries to get you more glucose. The way it does this is goes through a process called lipolysis, which means breaking down fat, but also making new sugar through proteins and glycogen. So if you start breaking down heaps of fat, what would you expect to see with the person? What would you expect, guys, if you start losing fat tissue? Yes, that's right. So you get weight loss. So that explains her thin build. This is a fairly common presentation with a type 1 case, particularly in children. They've had rapid weight loss or recent weight loss. That's because they're using fat as an energy source. Now, moving on, next clinical manifestation. As you start busting up all these fat molecules and sending it to the liver, your liver gets overloaded with fat molecules. And so it says, I can't deal with this anymore. I need to make a different metabolic pathway, which is ketogenesis. It's trying to make these fat into ketones. So unfortunately, ketones are acids. So we go into an acidotic state. If you start producing heaps and heaps of acid, guys, what do you think may happen to your pH? That's right, Lydia. So this is now going into the DKA side of things with diabetes. So what would you expect with the pH of your body or blood? Yep. So you have a big drop in pH, blood pH, which activates your respiratory center, increases its speed and rhythm, and that explains the Kussmaul breathing. Another thing that will happen is the low pH has a lot of hydrogen ions. Hydrogen ions really play a havoc with proteins. And then your neurons have a lot of proteins, so your neurons start to dysfunction. And as a result, you have a drop in GCS, or in her case, a decrease in responsiveness. And that was in respect to verbal commands. So there we have 4 done. I'm going to do more than 4 because there was more than 4 in this concept map or this presentation. But there you can see they're all separated from each other. They all don't come off just one point. And again, this is a very common way that students lose a lot of marks, is because they have all of these. So they could have lethargy, thin build, decreased responsiveness, and Kussmaul breathing all off cells can't make ATP. Okay, and that's why they lose a lot of marks, because they haven't gone to the trouble of explaining each individual mechanism. Okay, moving on. So once your liver starts breaking down all this sugar, or should I say other molecules, into sugar, we get even more hyperglycemia. Hyperglycemia is an increased blood sugar in blood. This amount of sugar is too much for the renal system to reabsorb. So the kidneys get overloaded with too much sugar to handle, and it goes, look, I can't get all this sugar back into the blood. There's just too much here. So that actually makes an osmotic gradient in your kidneys. So there's a whole lot of sugar in your nephrons that can't get reabsorbed. So as we know with osmosis, when we have big molecules, water follows them, and we start peeing out lots of water, and that's polyuria. This is the part of the reason why a person gets polyuria with diabetes, and that could be type 1 and type 2, but it's the osmotic diuresis. Another thing that happens is because you're passing out so much fluid so quickly through your kidneys, you're also losing a lot of other things, such as sodium and potassium. And this is one of the reasons for why you get vomiting with a DKA, is because of that osmotic diuretic effect, and it's the electrolyte imbalances causing a change in your vomit center in your brain which is leading to vomiting. The vomiting can also come out across here with the pH as well, but I've put it over here. But you would be right if you pulled it off to the over near the pH side as well. And then finally, because you're peeing out so much and you're vomiting so much, you get a lot of fluid loss, and therefore you're becoming dehydrated. The thirst center becomes activated, and then we become thirsty, and that's the polydipsia, meaning you're wanting to drink. Okay, so that's a concept map. Basically speaking, what we've shown here, that we've done a good pathophysiological process in the middle, starting with the etiology, creating a good sequential process. We've got a mechanism for each risk factor, and we've demonstrated a good separate mechanism for each of the clinical manifestations. So doing so will allow you to score pretty highly in your concept map section in your assignment. Okay, I'm going to shoot you across to Tracy now, who's going to take over with the written section. Tracy, do you want me to continue on this, or are you going to share your screen?
I'll share my screen if that's cool.